Good morning, everybody. I'm waiting for a boss. Good morning, everybody. Happy we're here in time, more or less. Today we will have a fairly packed program. But somebody asked me, we should present each other a little bit. Now, last year we made, but everybody put up his name, where he coming from, etc. Maybe I just will call up people, who are from different countries, so we can, so everybody knows where you're sitting. Why don't we start with France? In France we have two persons which are representing us, Philip and what's your first name? Sorry, no, yeah. So they are sitting here. Yeah, they are from the French Vivre avec une MPN. It's a very young organization, but very active. They have a very good thing, which is Septembre Rouge. They have achieved that in France, many official buildings, they have red color in the month of September. And this is something very interesting, and we're trying to make it a little bit more international, yeah. Then the biggest group which we have is a Spanish group, which I think today it's sitting around here. So we have about MP in Spain. We have many mothers here, which are defending the illness of kids, which we are trying to handle here. Then we have a Dutch organization, which is I'm sitting here in the middle. Yeah. We have this time a fairly large Swiss representative, just the line behind, yeah. And I think that's it, country-wise, no? Then we have our friends from pharmaceuticals. We have a delegation from AOP, Sophie, just sitting there. We have GSK. We have two ladies today. Hello. Hola, Ana. Yeah. What we have missing there? Nuestros amigos from our friends Morphosis. Thomas is there. He's a very, very pleasant person. We have beautiful talks to each other. And I don't know if the Novartis people is here. No. But I think that's probably about more or less. And in the back, the physicians, they always go to the backside. So it's, they're hiding there, And Ruben always, he works he gets his work uploaded. Who came in there is not the German. You see, in the past, the Germans were the most punctual. Not anymore, yeah. But he's one of our young MF patients, and in nearly all the MPN symposiums which we made, he was present, Thomas. So he's an old-timer here. Okay. We have a little change in the program today. We will have now first a presentation of Rao. He is a researcher and a very basic researcher. So he looks for new ways of, how can we attack our quality of life problems. And we had breakfast, but we will see a lot of dead mouses, which he is opening. And but he found a few very interesting things, which I like very much. He works in St. Gallen, Switzerland, and he will give his talk. Then after him, we will talk about the combos, which are the combined medicines. When the physicians will be here again, which I think is something which is very interesting. We would like to know these combos. Do they really have a future? Do they not have a future? And in the afternoon, we have there will be treatments about the anemia. And in which direction should research go? Especially in this research, I would also like to hear from you. Maybe you think it in the meantime. I will ask you, how would you see from a patient view about the future? What do you expect from a pharma industry? What do you expect from our physicians? And this is why we are always representative from a patients here. So I would very much think over what kind of questions you could put to the audience which we have here. And we will involve also today, we will involve the pharma people. I want them to talk and to explain, what are they doing. We're all working very hard on the products to improve the quality of our lives. So, but I need your cooperation, so that please, think it over that you are prepared, yeah? Good. More important people has to talk now. This is Rao. He's also called Tata. A middle name I cannot pronounce. Yeah, it's it's as complicated as mine. So please, I give a word to you. Yeah, here you have a micro.
I have it. My and here.
But put you on here.
The same like you here. So
they told me I'm the only one who gets one.
No, as you said, it's like a unique microphone. I have to adjust it. But thank you, Peter. Good morning, everyone. Peter, thanks again for this kind invitation and to keeping me in the loop of this exciting meeting. To be honest, I learned a lot yesterday, and then on the top, actually, it kept my motivation now too high. And after talking with yesterday, some of you guys, it was very interesting discussion. I think we need to do a lot more research in MPN. That's the message I'm telling now myself. So what I thought of showing you some unpublished data from my laboratory, and before that, I give you again a little bit recap of fundamentals of MPN. Yesterday we had a very nice didactic lecture from Laura. She introduced you what is MPN and how it evolves. So I go a little bit in that direction first few minutes, and then I go on and then discuss with you some unpublished and published data from my laboratory and my own experience, and I also a little bit touch why we failed to cure MPN so far and what needs to be done in the future. I add those stuff. So please feel free to interrupt me if you have any questions, and happy to answer you. Yes. All right. Just going back. These are my disclosures. The work I'm presenting today has nothing to do with my affiliation with my work with these companies. So I would like to start my talk by saying, or by asking you, how many of you like candies? How many of you like desserts? Can you please raise hands? Okay, I think it's not a bad number. So like the MPN cells do, right? So why I'm saying so, I will tell you why it's so important what you eat when you are healthy, but when you are ill, I think it's very important you should be mindful what you eat. Okay, so I learned and did one thing while preparing this presentation. Around 35% of all cancers or avoidable cancers are caused by diet. So this number is really means for me it's a huge, right? So it's in your hand actually how you can control your disease on daily basis. So I will a little bit touch why I'm saying this. So here I'm highlighting the diet. That is what you eat, what you are, right? So that is very important when you are healthy, but it's even more important when you have MPN, okay? So as Peter mentioned, I'm fully dedicated to find new ways, my career, find new ways to identify and target MPNs in a more meaningful way, if possible, to close to the curative approaches. So this cartoon you saw many times yesterday. So basically, MPN is not a single entity, right? It is it's a kind of, you classify like a caste, right, religion in subcaste. And so MPN is the same here. Based on the cell type increase, based on the symptoms, the patients show, scientists classified into 3 major flavors. One is P. vera, where you see more red blood cells. And the second entity, you have essential thrombocytosis, where megakaryocytes, more platelet production. And the third entity, we call it myelofibrosis. So the myelofibrosis can be primary. That means it's not coming via PV or ET, so it's a primary direction. And then you have other classification is secondary myelofibrosis, right? So that either come from the PV or ET can progress, that we call it secondary myelofibrosis. So both primary and secondary myelofibrosis basically share more or less same symptomatic features. There are a few tiny differences in terms of mutation burden, how fast they grow, and what was the previous history. But basically, primary, secondary myelofibrosis, phenotypically similar to me. And as you know, we didn't talk a little bit yesterday, this part. So all these entities, PV, ET, myelofibrosis, can eventually, at a certain degree, depending on what additional mutation that patient acquired. For example, if patient get new mutation in P53, so the chances of getting and progression to secondary myelofibrosis or PVET to secondary acute leukemias. So that is the last stage, is the deadly stage you can imagine. So all these entities actually basically arise. We talk a lot about it, and you are familiar with this because of changes in your gene, right? So those genes we already know. We talk a lot. It's one of the major genes that get mutated in MPN patients is JAK2. JAK2 kinase basically is the enzymatic activity it exerts in the cell, and that drive hematopoiesis in uncontrolled fashion. Yesterday, Laura nicely alluded that part. I'm not going too much into that. And because of these mutations in JAK2 kinase, and I pointed out here two mutations. One of the major mutations we often talk, and many patients have it, almost 80-90%, is JAK2 V617F. So what does it mean, V617F? It's just a single nucleotide in your DNA change that leads to a valine replaced by phenylalanine. So phenylalanine, valine, or amino acids that are actually building blocks of our protein. Now, because you have change in your gene, and then also you have change in the protein, and that change in the protein actually makes this protein structure so different. That structure makes this very constitutive, uncontrolled, without any trigger, your signaling, and then your hematopoiesis keep going. That's how you produce more blood cells. Okay. Now that is the major mutation, and subsequently, on the same JAK2 gene at a different place in the gene, people found mutation that is exon 12. Some patients, actually, these are not so many, very few patients, but this mutation exclusively drive erythropoiesis. No other phenotype patients, so no ET, no fibrosis. But it is a bit aggressive than exon 14, that is V617F. So biology is so interesting here. Same gene, two different mutations, two different locations. You have different phenotypes. And no one knows how different mutations on the same gene exerting different functions leading to different phenotypes. Okay, so that's one of the things we are biochemically we are trying to understand. If you know that why two different mutation cause on the same gene two different phenotypes, we can come in a new way to target those. Okay, so that's one thing. And then the second major mutation in 2013 by two independent groups, one in Vienna, one in Cambridge, identified this as the calreticulin. So the calreticulin, we heard, so by chemistry or biologically, it's a protein called chaperone protein. So the chaperones in our cells actually a kind of clearing or verifying mechanisms. If your protein is not so properly folded, it eliminates, basically. Now, this important protein is actually got mutated in patients, okay? Now, and these calreticulin mutations, actually two types. One is calreticulin 52. We say the type 1, when your doctor say, oh, you have CALR type 1 mutation, and you have insertion 5, that is type 2 mutation. Okay, again, there, not a hot spot one place. That gene is susceptible throughout, and then one mutation at deletion, other is insertion. But in this case, either deletion or insertion, both cause ET phenotype. Okay? I will little bit talk why only ET but not PV if you have CALR, right? And then those CALR patients, ET patients, eventually can also progress to myelofibrosis. Okay? So again, all roads leads to the myelofibrosis, either directly or via ET or PV. Now, you have a third mutation called MPL. So MPL is the myeloproliferative leukemia, as the name stands for, which is nothing but thrombopoietin receptor. So this is the gene actually present on your stem cells, blood-forming stem cells, sit on the surface, and then it waits for its partner. It's called thrombopoietin. Basically, it's a soluble circulating factor in your blood. And then when it touches this to the receptor on the stem cells, it activates the signaling, trigger megakaryopoiesis and platelet production. But now, because you have this mutation in MPL gene, and it is no longer required this stimulation via its partner, it itself can trigger alone. That's how you constantly, continuously produce. You end up having more platelets. Okay, both CALR and MPL cause essential thrombocytosis and myelofibrosis, but it won't touch much erythroid compartment. So the erythroid compartment mainly driven by JAK2 mutations. So that is the biology of this, okay? So what we are going to, so these mutations were identified, JAK2, almost 20 years ago, were identified. 2005, it was identified in Basel, Cambridge, and in Paris, and theoretically in 2013 and 2006 MPL. So why I'm telling these years? Because it's almost 20 years. We identified, we know which genetic modification drive these phenotypes, but we still don't have curative therapies, right? So it's a kind of a shame to scientists, for sure. We have to blame scientists and then clinicians. I think we are not going. I'm scientist. I take the blame. So one question, one possible reason I think why we fail to cure cancer, I think we are not looking at the right things, right direction, in the right time. Okay, so that's where we really have to invest. I will a little bit touch that part in my next slides. So this cartoon, it's my favorite cartoon, and because it came from my my excitement to do science in MPN. So what this cartoon I'm trying to show you, as I alluded before, that we are trying to find events that are changed early stage of your disease. To me, always as a scientist, late is too late. Okay, you need to you need to target your blood stem cells which are mutated in your bone marrow early enough. So what this cartoon is showing here to you is that, for example, we all have healthy blood stem cells. We call it hematopoietic stem cells in our bone marrow. When you are young and healthy, they function properly. And these are the cells, these are the precious cells are helping us to combat all infections and even cancers throughout our life, because they have two cardinal features. One is self-renewal capacity, meaning that they produce themselves like that, so that you have a pool reserve. And the same time with their second property called multi-differentiation property, meaning that these cells can give rise to all type of blood cells in your body throughout the life in a balanced, continuous fashion if you don't have mutation when you're young. Okay, so when you are aging, so you accumulate so much progressive changes in your body, inside the cell, outside the cell, including in your blood-forming stem cells. That already set a kind of a different tone compared to when you are young. So what happens when you are aged? You get more double platelets, even you don't have mutation. Okay, platelet number really goes high when you measure in your mother or in your child. Okay, so almost double. So that gave me some insights. We call it the myeloid bias. As the stem cells, when they get aged naturally, chronologically, they get a myeloid bias. Okay, so you can imagine why aged patient, if they have JAK2 mutation or CALR, the disease progression is really rapid compared to children. Okay, and we also modeling that in our mouse model. So we age mice when they are young and when they're aged, and we express this mutation, what we see in patients, and then we ask how kinetics of disease progression is really different or similar between young and aged patients. Okay, so but what I'm trying to show in this cartoon is that you have blood stem cells, and these blood stem cells by a bad luck or by accident you acquire at one point of your life. Right? So those mutations, I told you, JAK2, CALR, and then you also have additional mutations which are TET2D and MT3. So, and ASXL and so on. So the other mutations you don't hear very often. They are kind of set the base or a kind of secondary cooperating mutation. So these are epigenetic genes. So they do a different function than the JAK2 or calretically, but they also cooperate with their primary mutation. Okay, so those mutations you can also find when you do a next generation sequencing. Okay? Now, so whatever reason you have your stem cell get mutated. Okay? So this stem cell mutated, so how long you think that you have a mutation and you get disease? Anyone knows how long it takes to get a disease? It depends. But my answer is it takes decades. Okay? So in other words, you are diagnosed with MPN today, probably those bad events happened 10 years ago, 20 years ago. Okay? So it takes decades, it's a long wait. Okay? So we are now also asking question, what is really driving from after having this mutation? Okay? And why in some patients they never show? And the other thing you have to know is that this is no longer to me, MPN is a rare disease. Okay? Why I'm saying so? Because rare definition came 10, 20 years ago. That time we didn't have a good sensitive technology to identify how many mutant cells you have in your body. In other words, next generation sequencing was not so established. That time was very premature. Now, you can really fish out a single mutant stem cell or any cell, mature cell, from 10,000 healthy cells. Okay? So technology is so much improved. Now, with that technology, in Denmark, there's a big study. They measured almost 13,000 healthy volunteers to participate. Those who have cardiovascular disease, atherosclerosis, and other comorbidities, other problems, they all participated. They took the blood, they did the NGS sequence. So the surprise came. So what they found, out of 13,000, 635 patients have JAK2 V617F mutation. But those 635 never showed or never complained any or never diagnosed with a PV or ET or MPR. So that tells me is really that the number of patients with the mutation is more than the patients show the symptoms. Okay? So there are two things. It's no longer rare disease. I think it's many people getting it, and no one knows why. JAK2, please.
Of course, there is a difference between having a mutation and having a disease.
Absolutely.
So, although the mutation cannot be rare, the disease can still be rare.
Still be rare, exactly. That's what I wanted to say to you. Now, that is my next sentence, actually. So now you have more number of patients getting mutation, but not the same number showing the disease. So in other words, some people are blessed with overcoming the disease, right? Probably their immune system is good, their lifestyle is good, right? Or any other medication they are taking probably also helping. We don't know, right? But what you have to, the essence here, what you want to know, what you have to know is that getting this mutation is very often than getting the disease, okay? Now, if you know that why some people get disease, why others don't get, I think there are also some really important things you can learn, okay? As a scientist, I'm very much interested. Now, from no mutation to one single cell with mutation, and over time, decades, you end up in a diagnostic where you see a really detectable, from undetectable, from low number, very low number, to very high number, okay? In order to reach that stage, even though it takes decades, but this process requires a lot of energy, right?
In this study, they also search for the CAR. Sorry. In this study, they also search for the CAR mutation or just JAK2 mutation?
So in the first study, a very important question actually, the first study, proof of concept, they wanted to look how many people really have it. So they focused on JAK2, but now they also measuring in caloritically, general population.
Okay, thank you.
Mm-hmm. Now, so from an undetectable stage to a detectable stage, to reach that stage, you need a lot of energy to build those many cells, right? If you want to divide your cell, you have to give energy, otherwise it won't divide, right? So in other words, they really change their metabolism or the nutrient habits differently than when they are healthy, okay? So we think that to reach this process, as I mentioned, it takes decades. As I told you, this cartoon showing you, even some people have when they were born this mutation, okay, with this mutation. But they only show when they were 17 or 18. Yesterday we have heard 8-year-old, 17-year-old people have this. They probably acquired really when they are in mother or after the birth, okay? Now, whatever how much time it takes, but what are the factors that really bringing them from this low stage to the high stage? If we know early enough, and this disease, I have to allude that this disease comes from the stem cells, okay? If you are mature, let's say your red blood cell get mutation, you don't show that lifelong, only 120 days red blood cells survive, okay? If your platelets get this mutation, let's say they survive only 8 to 10 days in your body because you constantly get new, right? So that cell is, but your stem cell get mutated, that is the problem we have. That's why we call this as a clonal disorder of hematopoiesis, right? Now, if a stem cell get mutated, we need to better understand at this stage, okay? So much of the research focused on you take the blood and you measure, you do sequencing, you do all biochemical assays, and also new therapies. For example, all pharma companies test on cell lines in the cell culture system. Yes, you put more drug, they die, it is very effective, right? But that's where I think we fail. I think we need to really understand, zoom, where the problem come from, right? You have to see where the seed is actually planted, how it is different from your healthy stem cell. Okay, that's where I'm focusing my laboratory. I would like to share our some recent exciting findings at the end of my talk. And then also I kept the aging aspect, right? As we age, these mutations get, incidence of acquisition of this mutation is higher, and then also you have, on average, patients are more than 55, 60 years old. Okay, very rarely you find pediatric patients, we heard yesterday. Now, my laboratory asking if you know really what is happening, if you understand what nutrient or metabolic specificities of mutant stem cell versus healthy stem cell, what age-associated changes that are occurring inside the cell, outside the cell, you can go and attack those, and then and probably you can eliminate those stem cells. Okay, so that is what we are exactly doing in the lab. As Peter mentioned, that you see a lot of dead mice and dead things. Don't be scary to see those pictures, but I will tell you also how important it is to model first before you go into patients. And the mouse model is not like a tiny furry human, but they're kind of useful models. I will show that. But before that, I would like to show you one cartoon. So what you see in this cycle, so it is a very famous publication from Douglas Hanhanen from Lausanne. So he repeatedly pointing the characteristics of cancer cells. So we call it hallmarks of cancer cells, okay? So these properties, what was written on this circle, are entirely different from your healthy stem cells, okay? Now, so two I highlighted, we talked a lot about it. Sorry. Yeah, we talked a lot about it is and the metabolism, so and then also the inflammation, right? MPN is intimately associated with inflammation and metabolic changes. So these two properties are already considered as one of the hallmarks of cancer cells, okay? So my laboratory also studying how these two hallmarks of cancer cells talk to each other and then further drive the disease, okay? So that's also, I will little bit show you today the data that what we learned from studying these two properties. So as I mentioned, we first test our ideas, hypothesis, what is different between healthy and mutant stem cell using mouse models. So these mouse models were generated by Rageswara laboratory in Basel, where I initially trained on this disease, and then also others, where they engineered these mouse models by putting the mutation, what you see in patients, into the stem cell, into these mice. And there you can really controllable way, you can activate these mutations. Okay, they engineered such a way. And then you can appreciate here that this mouse model, like in patients, in the blue curve and red curve, those are the JAK2 mutation expressing mouse models, compared to the black bar is a wild type, so no mutation. Okay, so like in patient, you see a very big spleen. Within no time, these mice they show disease bit rapid than patients because their 100% cells are mutant. That's why you have a bit more rapid kinetic, okay? In patients, you have 20%, 2%, depending on what stage you have it, okay? That differs the kinetic of your disease. Now, you see the splenomegaly increased, red blood cell number increased, platelet increased, and then also they have a poor survival if you compare to the black curve to the red and blue, which are JAK2 mutant mice. Okay? So this cartoon tells you that mouse models in a way are very useful. They nicely mimic the patient situation, what you see, for example, splenomegaly, blood counts, and survival. Now, so use those models, we started asking our questions, what is the role of altered metabolism in MPN pathogenesis? Okay, so what you see, this picture is not so nice to see it, but that's how it looks if you open these mice. Okay, the first mice, what you see is the black one is the wild type mice, no mutation. They have a smaller spleen. I highlighted with the red box. But if you have a mutation, you see how the spleen looks like. Okay, so in both models, over time, it's 10 times bigger. It pushes the lung and heart and an uncomfort feeling, right? But what we learned, in addition to they develop these classical MPN symptoms, and I also highlighted with blue and red stars. If you focus there, so compared to healthy mice, where healthy mice have a nice fat distribution under the skin, we call subcutaneous fat, and then they have a gonadal fat about in the belly fat, nicely. But if you activate mutations, this fat is really, really disappeared, resolved over time. And if you see the same picture, same mice, after 2 weeks or 3 weeks or a bit more time when we wait, and then even you don't see the fat. So we call it body weight loss or fatigue or the cachexia phenotype, we call it, right? So that these mouse models also show such phenotypes, right? And then, but we were really wondered, how come we activated mutation in the blood stem cells, but we have problem in the systemic whole body, right? So how this is happening, and what is the really mediator or the secreted factors that come from your mutant blood cells and also attacking other non-hematopoietic tissues, in this case, the muscle wasting, fat loss, okay? So we systematically studied this aspect when after seeing these differences, you see now blue curve, red curve, the body weight. So when we measure body weight of these mice compared to mice with no mutation, they started falling apart. And then what we also found actually in these models, in the next curve here, showing glucose levels. Okay, when you measure the blood glucose levels, mice with the severe PV stage, late stage, your blood glucose levels really going down. Okay, and this holds true in patients if you measure glycosylated hemoglobin. So that is the indication of appropriate glucose levels in the blood. So in the severe PV stage, if you have 5 to 10 times more red blood cells than when you are healthy, so you get this hypoglycemic phenotype. And that was very interesting, and I will talk a little bit that. So that explains why people get fatigue. So why I'm saying fatigue here now, because red blood cells like so much of glucose, okay? They don't have a nucleus in their cell because when they mature from stem cell to the red blood cell, they spit out their nucleus, okay? Unlike other cells in the body. And they heavily rely on glycolysis metabolism, which is fueled by glucose, okay? So now you can imagine you have 5 times, 10 times more red blood cells, and you eat the same amount, but you have cells which are more dependent on glucose consume all the glucose, and you have no glucose available for your heart, for your muscle, for your brain, and you can really think now, okay, I'm not feeling good. That could explain why, biochemically, why you get fatigue, why you feel tired so fast. And if that continuously happening, and you also provoke other changes to accommodate or to compensate those problems, okay? And then the additional problems come. But over time, right? Now, we found that red blood cell use more glucose, and those glucose is not sufficiently available for your other tissues, which heavily dependent, including brain and muscle, and that's how one get fatigue, which we think. Okay, so that it's not only in mouse models. I come to the data of the patient data that we and others gathered. So then I ask question whether diet, what we eat, does it make a difference on MPN pathogenesis overall and also day-to-day basis, okay? And that also we model by exposing our mice. I told you before, mice with MPN mutations, they lose body weight. And then I asked, can we rescue this loss of body weight cachexia phenotype by feeding them high-fat diet? Basically, you eat more cheese and more bad food, okay? That's what we exposed to mice. And then you see now, compared to in the previous slide, that mice got a balanced diet we call chow diet in the research, and that has normal carbohydrates and fat and other minerals in balanced way. But when we expose the same wild-type mice with high-fat diet, you see they become really chubby. That's what you expect, right? That's what's happening. But the same diet, when we expose to MPN, you see now what's the picture. You can easily compare. Yes, they did gain little weight, but compared to healthy mice, they didn't gain weight, right? The more you provide, you feed the beast in you, okay? So that's what's happening. And you still have spleen size is the same, bigger, even bigger, right? Right? So now, you see, it's not only in male or female. We ask whether these differences are only specific to female or male. We systematically went and then checked the gender differences, and it holds true in both cases. But in mice, males do a bit different than humans. Okay, so it's a kind of evolutionarily, there is some difference between mice and human in terms of gender when you look metabolic aspect. But overall, take-home message is that it's true for both male and female. Okay. Now, after seeing this, we also ask, okay, overall picture look like. What about blood counts? So here comes the surprise that when we expose mice with the food that has more than 85% fat, okay, that's what we did. And then in this case, we modeled calreticulin, mice expressing calreticulin mutation, and then we measured blood counts, including red blood cells, platelets, white blood cells. Since calreticulin mutation drives exclusively platelet phenotype, you can see within no time, platelets so increased when you expose them to high-fat diet. Compare this red curve with high-fat diet, black curve with balanced diet. So, and then we really went on and then went to the details why this is happening at the cellular level, biochemical level. I can tell you in one word that imagine a big cell megakaryocyte. One megakaryocyte on average in humans can give rise to 600 platelets on average. Okay, and the platelets are nothing but the clones, they shed from like a balloon, small balloons, packed vesicles from megakaryocytes. Okay, and that number heavily increase when you expose to high-fat diet because these platelets are really packed with fat. They are just membrane, and this membrane is full of made with fat biochemically. Now, so you provide more fat, they utilize it to produce those tiny balls, and then you increase. So this is one tiny information from these small preclinical models of MPN. You see how even just, they have the same mutation, you just expose them to different diet, and then it fits. Okay, so this is one example how important it is. You think if you have PET, what you should eat, what you should. I'm not a cancer nutritionist. I should not advise anything, but I advise you to consult your cancer nutritionist. He or she can advise together with your physician. Okay. Now, you might be wondering, Rao, I have PV, I have JAK2 mutation. So what I should eat? As you know that PV patients have more red blood cells, 3 to 4, 5 times more red blood cells. As I told you, red blood cells like glucose, like we all, okay? Now they like even more, but they not only use more, they also use differently. That's also we learned, okay? I'm not going to that part, but if you have any questions, if you need more information, I will be happy to provide. Now, in this case, so we switch to JAK2 model here. Right? JAK2 models, I told you, they exclusively show PV phenotype, red blood cells. They have 5 times more red blood cells. Now, we expose them to 8 to 12 weeks with a normal diet, but in the other group, we expose to high glucose diet. So it's more than 65% carbohydrate, it's a fructose, sugar, sucrose, or glucose, the composition, whatever carbohydrate, that in the end, it leads to the glucose. Glucose is utilized, your cell taken up. And then you can see now in the pictures there, the first bar graph, what is showing, black corresponds to wild type no mutation. They have no problem. They have a balanced way to utilize it. They don't utilize too much, right? But now it came to the surprise, even at stem cell level, we found when we exposed the JAK2 model with high glucose diet, stem cell number went down, okay? Number went down transiently, but that number, they went down because they're pushing towards the erythrocytes, erythropoiesis. That's why the next subsequent stage, where I labeled it MPP, multipotent progenitor, that is one step down of your hematopoietic stem cell. If you put a hierarchy, stem cells stays first, and the next step is MPP, that is the uncommitted. And then you have, I call it pre-CFUE, these are scientific terms, colony forming unit erythroid. So basically that is the progenitor cell, stem cell, destined or decided to go to erythropoiesis. No longer it has a multipotency. It can no longer give other cell type. Okay? Now if you look at here, it start picking up already at stem cell number within no time, 8 weeks, 12 weeks. Okay, imagine patient constantly taking too much dessert, too much sucre, too much beverages, over time 10 years, slowly every day it pushes erythropoiesis, slowly. That's what you see, it progressive changes. Now, when we also measure reticulocytes, reticulocytes are nothing but one stage of the mature red blood cells. They are just freshly generated. They're not old 120 days living, just came from stem cells. Those are reticulocytes, which you can also measure in your diagnosis. You see again, IFAD diet really picking up, even within no time. So that's scary to me, that, okay, you feed your cells whatever they want, and then you complain that I have more disease, my treatment not working. Okay, treatment is one side, but also something is in your hand. What you eat daily, I think you should really think what disease you have it. This information I'm giving everything from mouse models that we gathered. Okay. Yes, please.
In this case, you're focusing on exon 12?
We also did on JAK2 V617F as well.
And that's the same result?
Similar trend.
Okay.
Similar trend. JAK2 exon 12 show a bit more faster kinetics, but just for presentation I picked, but we have all data on JAK2 V617F as well. Now, so I just wanted to show you a little bit how we can take this information for a therapeutic purpose. In other words, I ask a question, is there any therapeutic window on metabolism-based approaches? Okay, based on our findings and findings from other people in the field. So for that, we did a very detailed molecular analysis. So what you see, the form of heat map here, we call it. Yesterday, Ruben nicely alluded that similar cartoons, where you have blue is low gene expression, red is high gene expression. Okay, where we took the stem cells from the mice with no mutation, mice with the JAK2 mutation, and we systematically look the molecular changes by a technique called RNA sequencing, which is similar to next generation sequencing you do for mutation profiling. Okay, now, and then we listed out the genes, what you see, those PKM, PKFB3, those all genes are very important in our normal cells, but they are heavily upregulated. In blue and red, you see E12 and V617F. If you look compared to wild type, blue means no expression or very basal expression, which we needed, but they massively upregulated in the red, right? So that facilitates, that means after expressing mutation, your cells are changing molecularly something, and that facilitates to use more glucose, okay, more glycolysis. And then after seeing that such a nice pattern, we focused on, can we use this information to target MPN cell growth, to reduce platelets, or to reduce spleen size, to reduce erythropoiesis. So in this project, what we looked into it, one of the genes that caught our attention is PFKFB3. So this is the rate-limiting gene of the glycolysis. Basically, what it does is that it speeds up your glucose utilization. Okay, if you have more of this enzyme in your mutant cell, it speeds up the glucose process to the end product. That's how they get energy faster. So that's what I meant. They not only take more, but they also utilize really differently because of this molecular wiring. Now, after all our efforts, that led to identification of some important genes, PFKFB3. So fortunately, there is a drug that can inhibit the activity of this molecule. Okay? As a proof of concept, in mouse models, we tested what if you inhibit the activity of this differently present enzyme with this drug, can we modulate the disease progression? Okay, that's what exactly we modeled. I showed you in the cartoon here. So we exposed the same drug with no mutation, with mutations, mutation 1 and 2. So blue graph showing the spleen size, okay? So what we did now, we didn't treat V617F red in the exon 12 mice with no treatment, we call vehicle, it's a placebo, okay? Now, and then we treated this with this drug V1, Warburg effector inhibitor 1, we call it. It's for historical reason, and biochemical, Warburg is the guy who actually found cancer cells utilize glucose. Even 100 years ago, without any of these tools, fancy tools. Okay, this was forgotten almost 80 years. Now, since the last 20 years, again, people dig the literature, and then actually, holy people identified this important information a long time ago. Okay, now people focusing on this, including us. Now, you can appreciate here, when you treat with our inhibitor in JAK2 models, you can really reduce within 8 weeks. We didn't treat too long, but if we treat too long, we can really maintain it. It is tolerable if you reduce the glucose utilization with this inhibitor, but you can see that spleen size is really reduced in both models. And we also combine that with the classical JAK inhibitor, if it can further make it better. Okay, in a combination therapy. Okay, so in exon 12 model, it is very striking. In the last red curves, you see it when we combined our inhibitor with JAK inhibitor, Ruxo, the spleen size is really getting back to the normal size. Okay, so that was very exciting to us that our hypothesis that indeed metabolism really playing a role in driving disease and inhibiting metabolism, you can also alter the disease progression. So you might be wondering how metabolism is important for all normal cells, normal function, glucose also important. How can you target in patients? You probably end up with side effects. Okay, time will tell, but my answer is here, you are going with a specific target that is not expressed in your hematopoiesis. If you look at this gene that I put the arrow here, it is not expressed PFKFB3 in your normal healthy cell. Okay? It is only turned on if you have mutation. So I call it they are really a specific targets. Okay? Whether how much it is specific, time will tell. We need to do more work. Okay? And then with the approach, we don't want to really completely inhibit. We want to bring back to our healthy level. That's it. Okay? I'm not here saying not to completely to make it zero. Yes, if you make it zero, other cells also get affected, right? You bring back to, you tone it down. So when you tone it down, even mutant cells no longer have the fuel to proliferate, to differentiate. MPN is nothing but a number game, right? We talk a lot. Okay, I have 500, 600, 13, 20, right? Depending on what parameter you're looking. So this number game is fueled by what you eat. That's what I'm trying to say here. So be mindful what you eat, which disease you have. So then I was very satisfying to see our data was really confirmed, mouse data, what I showed you, by people like Ruben Mesa, actually, this study from one of his colleagues, and he in his department, what they showed 1 year after we did the mouse work, they exactly came with a very nice data from inpatient cohort. What they elegantly did, they took a cohort called NIH AARP diet cohort. That was a very big study in the United States, recruited 450,000 volunteers. And then they they did a questionnaire, what they eat, how much they eat, and in that study, systematic study, out of this number, they found 500 people are with MPN. Okay, out of these 450 people. Now, these 150 of them ET, 190 of them PV, and others probably myelofibrotic. Okay. Now, you see, general population, already you have a 500 MPN. And then what they did, they classified based on how much carbohydrate they eat, in sugars they eat on daily basis. So here comes the surprise. So very satisfying to see that what we found exactly in mouse models, they did very unbiased study here they found. So what this cartoon shows here, maybe I go a bit closer. So here, this line tells the patients with 70 grams per day sugar they took, okay? And then these patients actually above 120, okay, within MPN patient, 120 grams versus 70 grams. So low sugar, high sugar, okay? Now, here the statistics telling you hazard ratio, okay? So hazard ratio here, very significant means the chances of a disease progression is higher. And then the phenotype they show is very higher if they took the more than 117 gram versus 70 grams. So then you might be wondering, Rao, sugar comes from what? Chocolate, milk, fruits, right? All contain sugar, right? They also systematically looked, okay, whether how much fruit they take it, whether sugar come from natural source, sugar come from beverages, or the fruits fructose, okay? Irrespective where it come from. They found a statistical, meaningful, a significant way that if they take more sugar, they have more chance of increasing the disease progression higher. So, okay, so this is again a very nice comparison. We looked into mouse models, and then they looked into patients in unbiased way. Two independent studies. No one knows each other what they are doing that time, but they come to the same conclusion. Okay, so in other words, you really need to think what you eat when you have a PVET or myelofibrosis. Now, so, and what about fasting, you know? So during festivals, because of religious festivals or ancient people, actually, they did the fasting without knowing its benefit, but they probably felt benefit over time to lose body weight or to clear all your bad things in your body. And we also exposed our mice with a fasting, meaning that we restrict the calories there with the food. In the food we designed that is less calories, and those less calorie diet, we put them 12 hours food, 12 hours no food, not continuous exposure. So we did several cycles like this, and then we ask how it affects. Here comes the surprise. Again, so your JAK2 model we used, and then the dotted lines in the red you focus, that is with fasting. So food on and off. And then the continuous line, the red line, it's a continuously we fed. Okay, that's what we do regularly. And you see the difference, mice with the same mutation, we have fasting. Okay, less nutrition availability, less disease progression. But again, you might be wondering, Rao, I one side complain fatigue, I have no energy. Now you suggest me that fasting is useful, right? Again, so it depends on what you eat, not that how much you eat and what you eat, I'm trying to say. Okay, you should not completely not eat. Okay, you should eat to maintain your healthy routine. But what you eat, how much you eat, it is very important. Okay, now it not only reduced blood counts, but it also reduced spleen size. That was very surprising, because all increased blood cells, you have a bigger spleen, right? It was a kind of fitting. Now, I just wanted to summarize what I told you, right, in this cartoon. So here you have stem cells. It gets mutated. Mutation leading to molecular changes, including changes in your glucose transporters into your cell. Okay, any nutrition, glucose, fat that enter to your cell via receptors present on the cell. Okay, I highlighted in the blue. And their level of expression increase if you have mutation because of molecular change. Together, they work together, they take up more glucose, and that energy facilitates to produce more blood cells. Okay? Now, what happens if you continuously utilize your money? You don't have any more money, right? You depend on bank to get some debit, right? Bank is a fat here in your body, right? So that is exactly what is happening. I showed you in my first cartoon that mice even lose body weight. They lose fat. So what happens that when they reach to produce a lot of cells over time, they don't have any more energy. If you don't eat it, you don't provide. They search for the alternative source, which is fat, right? So that's why fat becomes shrinking. So you lose over time the fat, okay? So that fat is you lost, and then at the end you don't have fat and you lose the body weight, right? And also eventually you lose muscle weight, right? That's where the real cachexia phenotype comes. So how do they do it? How mutant cells go and contact bank to get the money, in this case fat. So they secrete some inflammatory cytokines. So again, inflammation come into play a role. So how inflammation, we talk a lot about inflammation increase in MPN patient. What it really does, right? This is one of the things it does, right? It go and stinch fat release, it's a stored energy. Fat is nothing but full of energy storage, right? That they release, and then your cells utilize. And if that continuously happening, your bigger fat cell becomes smaller fat cell. right? And then you lose the fat. That's how you get the cachexia phenotype. That's how you lose the body weight in cancer situation, including in MPN. And this happens more in severe PV and severe myelofibrosis models. We also notice that, and this holds true in patients. Okay? Now, so this is the mechanism. Any questions so far, if anything? Okay, so if we can also discuss later if you have any questions. So what I'm going to tell you now, how we take further, how we take further this information to make it more beneficial to the patients. Okay. So the cartoons I'm going to show you next 3 slides I got from my good friend of mine, Angela Fleischman. So she's also clinician. She worked a lot on MPN. She's California-based researcher, clinician. And what she did after studying our research, publication also from Ruben Mesa, and she initiated a program. She recruited almost 50 MPN patients, and then now she wanted to keep them on a controlled diet, like we did for mice. Okay? Now, and before that, I like to show you this publication, landmark paper, that came from actually Spain. Okay, Valencia, Barcelona, Madrid, and all collaborators did on cardiovascular disease. Okay? As we know that if you have heart problem, you always think about fat, right? Why can't we think the same in MPN? Right? We now know that fat feed platelets, glucose feed erythropoiesis. Something else feeds myelofibrosis, okay, which we are working. Now, so this paper really stimulated many researchers, other cancer researchers, to do the same thing in different settings. So what they did, they simply exposed patients with Mediterranean diet with compared to Western diet or their natural diet. Okay. Now they found really good beneficial effects if patients under Mediterranean diet. That's what Angela Fleischman did. So what is Mediterranean diet? You guys know better than me, right? So it is basically, if I define that, it's a simple plant-based diet, balanced diet, fiber-rich, and it exerts anti-inflammatory properties. Basically, it reduces inflammation. Because of more fibers, then you ask how fibers can help my metabolism. It gives the beneficial effect. The fibers, basically, when you take, it kind of put a layer in your stomach, okay? And that layer is important for the microbiota, what you have in your gut, in your stomach. And also, it slows down the glucose uptake. Okay, even if you take the same amount, which is not recommended, because it is coated with fibers inside the stomach, and it slows down the absorption of your glucose. It's not pushing so quickly. Okay, and that trigger is slowed down. You are not hungry, and then also you feed your cells a bit controlled way. Okay, that's how fiber-rich diet helps. Okay, one way to microbiota, one way to slow down the utilization of your sugars. Okay, now the similar test. This is the regime Angela Fleischman did in her clinical cohort. She recruited 31 patients in the first study, and now she increased to up to 50. She divided them 50-50. She exposed them to Mediterranean diet and the US diet, I would say, Western diet. Now, here it comes the surprise. What she found here, very interestingly, she followed up to 15 weeks these patients, and they really strict to adhere to our suggestions. They did exactly what she asked to eat. And then she compared the black bars or the no control. They can eat what they want. But in the pink bars, you see that it's really falling part of the signature MPN SAF score. So yesterday, Laura and Ruben nicely alluded the SAF score symptoms. So how you are feeling today, whether you have any bad mood, whether you're social, anxiety, fear, night sweats, all those stuff they counted, and then they found over time, compared to normal diet people, 17 versus 47, 31 versus 53. These looks like a tiny difference. You know how much difference you need to feel bad and good, right? It's getting to the positive side. Why can't you use that in the form of diet, and then the top you add yoga, meditation, and other good things along with your medication. I think we are getting there. We are not there yet, but we need studies like this more in different places to see how reliable it is, how useful it is, and then we can really find new ways to modulate the disease. Now, So this is the study from Angela Fleischmann lab, and we talk a lot about thrombosis. Martin yesterday nicely alluded, so how it evolved thrombosis when the platelet gets activated. Thrombosis comes, right? And in ET patients, you not only have more platelets, but they're also very sensitive to if you give any trigger in the form of collagen exposure or DNA exposure or inflammatory exposure, when they touch to neutrophils, they kind of activated, they release these factors. That's how you get it. So, but what she also found that she connected to metabolism. So MPN cells use more metabolism. What happens if you use more? You produce more waste, right? And one of the waste in our cell is called reactive oxygen species. So those are oxygen molecules like a fine sharp needles, and they can go and attack all the parts of our cell, components of our cell. Okay, that's how they cause the activation of platelets. If that is happening in platelets, that provoke the thrombosis. And then what she found that so she came up with the antioxidant, which is called N-acetylcysteine, which is the very old drug. Basically, this drug reduces the ROS levels, basically. Okay. That's if you reduce ROS levels, okay, even if you have more metabolism, you can reduce thrombosis. Okay, that is very nice study in vitro, in the cell culture models, in mouse models. Now it needs to be tested in patients. Okay, so why I'm bringing this? How important it is that metabolism actually the major culprit. Okay, to produce more number of cells. And then you have more cells. You always have to think your number. This number coming because you're feeding it. Okay, now. So I switched a little bit my gears to a new thing. So Peter asked me to also a little bit touch what is new in MPN. Okay, so what I told you is the past, and that is the information I shared with you from our data colleagues from in the field. But now what is new? What else we can bring and what is missing? Okay, I'm going to share with you. It is a bit confidential. Please don't share with anyone. This part of the study we now submitted to a manuscript in Nature Cancer. So two questions I ask as a scientist, okay, why and how. So in this case, how EVA mutations expressing in blood stem cell causing entire problem, body problem, systemic problem. So what are the factors that connecting blood stem cells to the systemic part? And then second, I ask, can the systemic alteration can be harnessed? Can we leverage those changes, those findings to find new ways to target? And even to find the biomarkers. So yesterday, our discussions, Ruben Mesa, he also mentioned we really don't have a good biomarkers. Okay, and also one of the MF patients, they said that they were wrongly diagnosed with ET, even though that patient have MF. It's such a sad thing. So still we don't know how to distinguish in an easy way and in a reliable way. Okay, so that part I'm going to touch now. So very exciting data we discovered for work from last 4 years. Very extensive work. This cartoon showing. So what I did here, I no longer stick to mouse models. Now I have a very big access to MPN patients. Okay, so patient plasma or blood cells or bone marrow aspirate. So you can see the number, actually, the number of patients that I have access now. I collaborated with different teams, basically, okay, all over the world. And together with our mouse models, combining these two important tools, and I did all possible molecular biochemical assays, okay, to really distinguish what is similar between mouse models and MPN patients, and how they are different between when they are healthy, okay? So the ultimate goal is to find new targets, new biomarkers, and then use them for therapeutic target. So I'm going to share with you all our experts led to more than 1,500 reliable candidates. We cannot study all 1,500. We have to focus on few things, and then we focused on for several reasons. I'm going to show you a factor called GDF15. So GDF15 stands for growth differentiation factor 15. So it's kind of one of the inflammatory molecule. You can put it in simple words. So it circulates in your blood. So what we found that this molecule basically, both in mouse models and patients, is one of the really reliable, faithful candidate is very heavily upregulated. If you look at the graphs, it's the time kinetics. We measured in the blood of this molecule, of the red and blue are JAK2, black is wild type, no mutation. As the disease progress is speaking up. Okay, 10 times more in exon 12. Calreticline is the same, exactly. I didn't show in this cartoon. I need to update for presentation. But again, in the circulation, you have so much GDF15 in mouse model that anti-correlated with the body weight. You see the next graph showing that if you have more GDF15, you have less body weight. Okay, so we have a factor now that defines why patients lose body weight. Okay, why they lose cachexia. Now, measuring those, and then you might be wondering, Rao, show me the data in patients, right? So that's what we did exactly, the lower group here. The first bar showing the healthy controls. We have 55 healthy controls who has no hematological symptoms, no MPN, no other blood problems, okay? Now, and we looked into PVET, secondary myelofibrosis, primary myelofibrosis, even triple negative patients, a large number. You see, not like in mouse models, in patients, you have a continuum, but you have a really significant and 10 times higher increase of GDF15 in their blood. Okay, this was very surprising to this result to see us even in patients. And then what we found more interestingly that myelofibrotic patients is way high because it is progressively accumulating. It makes sense, right? Myelofibrosis, you call it kind of after the PVET or at the late stage, or even those cells produce more. But at the end, myelofibrotic patients have so much higher, 10 times more higher, and that gave me these results gave me one question, Rao, can you use this to find myelofibrotic progression at early stage? And then, in other words, I asked, can we use this as a biomarker to predict myelofibrosis evolution? Okay, and then that consumed a lot of my time.
I expect that those patients you have as reference group also have medicine.
Absolutely.
Yeah.
Why you see?
What is the effect there? Yeah.
Absolutely. We also extensively did how ruxolitinib affect, how interferon affect. We have all this data. For the sake of time limit, I'm not going. If you are interested, I will be happy to discuss that part. Yes. So one question we asked, this increased GDF15, it can be used as a biomarker to predict myelofibrosis. As you know, myelofibrosis is a diagnosis very painful. You go with thick needle, you insert here, you aspirate it, and the chances of getting the bone marrow into your needle is even low if you have fibrosis, because your tissue is not filled with the marrow anymore, right? You can get it from PVET, but in myelofibrotic. So that painful procedure, and also we don't know when patient actually progressing to myelofibrosis, okay? I thought it's very important to understand using GDF15 as a biomarker, and we did some computation stimulation. So what you see here is the curves. In the clinic we do these curves is called receiver operating characteristic curves, ROC curves. Whenever you heard this term, that means you are defining the specificity, sensitivity of a marker that you identified. How specific it is to your disease, how sensitive it is to predict as a biomarker, okay? Now, that's what we did. Upper is primary myelofibrosis, lower is secondary myelofibrosis patient group. And then what we did, we set a base 190 picogram that you find in healthy people, okay? And then asking question how this curve falls, okay? And if the curve is really straight like you see it here, we call it area under curve, AUC, when it is close to 1. And it gave very good sensitivity of 94% in PMF, and even secondary myelofibrosis, 100%. Okay? So this gave us a confidence that we can really use as a predictive biomarker when ET or PV patient progress to myelofibrosis. Okay? All you have to do is not the bone marrow, maybe bone marrow in the first case, but take the blood. It's very easy, right? Instead of bone marrow. And then you run this assay to measure GDF15. Okay? Then you can really measure it. Now, these are computational predictions. Okay? Now, Rao, show me the data. whether it is true. So, and then we went on and collaborated with in Canada and in Austria. Same patient when they were at ET or PV. So here I'm showing out of 33 patients we have PV, patient 1, patient 2. When they are at PV or ET, when they transform to the myelofibrosis after 3 or 5 years later, we also have access to the bone marrow from the same patient. Okay? And then we simply did expression analysis for GDF15 by a technique called immunohistochemistry. That's what we people do in pathology and diagnosis. And then what you see in the last, I don't know whether it's good on the screen, but red dots on the last part, okay? Last stage. So that is corresponds to the GDF15 expression in bone marrow. Out of 33, all 33 patients, irrespective whether they are Ruxo or interferon, when they progress, they have this higher expression of GDF15. So this gave us a confidence that you can indeed faithfully use GDF15 as a non-invasive. I'm saying that no needle insertion is required anymore. You take the blood, you measure. Of course, you have to rely also on other parameters what we have, and then combine those to get a nice conclusion, not to get confusion whether it's ET or it's MF. And the other advantage of doing this, having these biomarkers, clinician can also decide and adjust the dose or the treatment type, okay? So that's also, this is very, very useful information. And then I would like to tell you two other exciting in the next 2 slides, where the GDF15 is just a cause or consequence, because you have more mutant cells, you have more production of GDF15. And then we also ask that question, whether it is a consequence or a cause, okay? Whether, in other words, it's also promoting the disease. Okay? For that, we switch back to our mouse models. They are very useful, where you can modulate a lot. You cannot do anything in patients at this stage. And where we did was we genetically knocked out. So in these mice, you have mutation of JAK2, but no GDF15. Okay, and ask question, how disease progress in these models without GDF15. And you can see in the cartoon body weight, I told you, JAK2 exon 12 mice, for example, severely aggressive disease, it leads to body weight reduction. When they have JAK2, you lose body weight compared to a black curve here, wild type, no mutation. But here in the cyan color, you see it, JAK2 model without GDF15, it even increased the body weight, right? You really can reduce the cachexia phenotype that is coming from you because of your MPN mutation. This is very, very striking. Now, what about hematological parameters? Indeed, actually, it also reduced spleen weight within 6 to 8 weeks after removing, and it even improved the survival. It extended, when the mice have the JAK2 exon, they can only survive 50 days. You can extend to 75 days. So that's a very good improvement. Now, you ask, okay, I don't have JAK2 exon 12 RAW, but I have JAK2 V617F. What about the JAK2 V617? So we went on, also did, we knocked out the gene. It takes 1.5 years. This whole cartoon work took almost 1.5 years, okay? Because to genetically manipulate, your mice should have JAK2 V617F mutation, same time it should not have a GDF15. So this technically takes even more than 1.5 years. But it was worth, you can appreciate here now, when we knock out GDF15, surprisingly in JAK2 models, it didn't touch much of the erythroid compartment. It really lowered the myeloid compartment, where neutrophils, white blood cells, monocytes, you see the blue versus red curves in the bottom panel, really reduced. But very strikingly, what we found is that the platelet number on the top right there is really blue versus red. Blue is with mutation with more GDF. Red is with mutation but no GDF15. Okay? The platelet number almost 50% reduced within 20 weeks when we followed these mice. Okay? That is very striking. I never saw any drug reduce that much, including Jakavi or interferon. Now, okay, Rao, I have a calreticulin. What should I do? Right? And that's also we did. That again, in parallel, we knocked out in calreticulin. We got the same picture for platelets. And it has no effect on, anyway, CALR people don't show any red blood cell phenotype. That makes sense. And then the myeloid compartment didn't get affected, but platelets again. Really 50% reduced. It's not just in one model. And in all models we assessed, It's genetically, it's very conclusive data. We think this could be used as a not only biomarker. In future, we should really inhibit this molecule in MPN patients. So that's where I took a very big step, invested a lot of time to develop an antibody to target GDF15. Because you have too much, I want to inhibit that in patients, okay? So this is the antibody we generated. We tested in mouse models, both calreticulin and JAK2V617F, where we injected human patient-derived cells into mice. We call it xenograft, and where we followed systematically with our antibody, without antibody. So you compare now for color, for example, pink versus cyan, okay? Within 4 weeks, these cells really grown so much in these mice, human cells, whereas when we treat the same mice with the same cell with antibody, you can really reduce. That's exactly what we found in mouse-to-mouse experiment. So that means this, indeed, you can pharmacologically inhibit your GDF15 in your body with antibody or a different pharmacological approach. We are also now currently pursuing it with a different approach, not only antibody. Okay, so but this is unpublished. We recently submitted to manuscript, and hope reviewers see the same potential that we see it. Now, so one final or two final cartoons I would like to show you. Peter asked me, Rao, so which direction we should MPN research go? Okay, so what else missing? To my knowledge, to my understanding, we know nothing about the true things happening at the stem cell. We are just started, I think. We need to do a lot, as we discussed yesterday. So that explains why we are failing to cure cancer. Okay, I would like to give 2 slides insights why we are failing to cure cancer. So one problem is resistance. Okay, resistance, like if you give antibiotic, is no longer working because your bacteria is not responding to that. Same likewise here, your cancer cells not responding to your Ruxo or interferon. Okay, but that also after prolonged treatment. Okay, resistant is one problem, and then other problem is you have to look carefully. We are talking about JAK2, calreticulin, MPL. All these genes, when you are healthy, they are very important genes to keep your hematopoiesis ongoing. Okay, now such important genes get mutated. Okay, now you want to target those mutant genes, right? With ruxolitinib. Ruxolitinib is nothing but JAK inhibitor, right? How can you target an important gene to cure your MPN, right? Although JAK2 gave a lot of information to how disease is happening, but you also get side effects, right? If your drug is not specific to your mutant JAK2, right? And then it also hurts your healthy wild-type JAK2 that they coexist in the same patient, right? So that is one other problem. And the other problem, genotype, phenotype, I call it so you have additional mutations, okay? You target JAK2, but some other mutation also, you have it, that also contribute to not to be effective, okay? And then as we discussed, also politics, right? But that is a different topic. Now, so in other words, you really need to understand what is similar, what is different between your healthy stem cell versus your cancer stem cell. If you really tease it out, I think you will really find a good targets to target your MPN to achieve molecular remission. And then one thing I wanted to highlight actually, in this context, we are also working on now, we have a signature, molecular signature that we identified is at the stem cell level when they have mutation 1, mutation 2, or combinations compared to healthy, and then you come and attack those molecular signature which are presented only on mutant cells but not in healthy cells. Like this concept, many people working, but now we have our own approach, we have our target, and hopefully I can show you in the next meeting the outcome of this study. And with that, I would like to say one final thing. This is one of my famous cartoon again. So we discussed yesterday, and Professor Ruben Mesa nicely alluded that these patients, in addition to show classical MPN symptoms like increased blood count, splenomegaly, they unfortunately suffer from several oppressive symptoms, which including inflammation, fatigue, depression, night sweats, all cumulatively cause patient life is lead to the poor quality of life. So one question we are addressing actually, how these mutations in blood stem cell cause poor quality of life. Like we studied fatigue and why they get bad mood, why they are nervous, why they are anxiety, why they have anxiety, why they are depressive. So we are also working in that context, how this inflammation, how disease actually over time cause these undesired poor quality life. So that's what we are working now, and with that, I don't want to bother you, but I would like to end my talk by saying that we need more research, and MPN to me is like a hidden pandemic, right? It's like a COVID. When we have COVID, we all came together, we all left right, and then suddenly vaccine came. I think that much rapid and involvement we need from research side, clinician and patients, and I think we need to really. And I would like to invite you for a 30-day sugar-free challenge. It's not a ice bucket challenge. We should really test this in MPN along with this. And I would like to end my talk by saying, acknowledging my team. I have a small team. I don't have a very big team, but I have wonderful collaborators who provided me all clinical samples at our center and all over the world, and my funding support. With that. I would like to also take this opportunity to invite you all, save your date, and I'm organizing a MPN meeting in Switzerland next year, May 14 till 16. It's a beautiful place in Galen. Professor Ruben Mesa is one of the key speakers there, and Peter also coming, and we included a patient voice lectures like we are doing, and almost 40 speakers, 3-day meeting, heavily focusing on MPN. I would like to invite you all, please join and save your date, and thank you for listening.
So maybe we can make our next year symposium in St. Gallen, yeah?
Please welcome.
It's a gorgeous city in the east part of Switzerland. I think has 150,000 inhabitants. Old-style city, beautiful. It's a dream. So maybe we make our MPN symposium spring meeting. We make it we will see. Anyway, for me it was really enriching to find Rao, which we came together by chance, and I think it's important for us to know where is really the new products, where are we starting from, and without people like him, I don't think we would have new products. Poor mouses, by the way, were coming from Barcelona. It was a big mouse factory in Barcelona, actually owned by one of the big professors. And where all the poor mouses are being produced here. So in Catalonia, we're not only producing pigs, but we're also producing small mice, yeah. Anyway, so I'm very happy that he was ready to come here and to explain to you. I know it's very heavy for a patient, to hear all these things, but I think he managed to bring it in a fairly patient-friendly manner. I have seen his first presentation, and it was a bit different.
I didn't know the concept here, who is the audience, and then once you mentioned, then I a little bit...
And it's... he's also a fighter. He's a young person who is fighting for his future. The big problem for this kind of people is money. The financing is a huge problem. They have an easier life in the States. In the States, there's a bit more. It's not freely available, but they have more money available than here in Europe. Europe, unfortunately, for research, is not very. They don't have so much money available, so he is desperately looking, for financing, and maybe we can find ways together. We help each other. I know a few people. I'm also addressing myself to the pharma people, which is in here. I listened to what he's saying. We have to support these people, because if they are not doing their job, we will not get new products. I mean, he has been showing things so clearly in the results, and he is working, on since several years on these aspects, and he's one of the persons who really concentrates on the side effects, tiredness, etc. So it's very important, and I thank you very much that you have been here for all your work, what you're doing. And I did to think over some questions. I would like to have one of our physicians. What is your relation with people like this? I mean, how do you see the work which we are doing and what he presented here? Do you consider that this is going to help, in your work? I don't know who of you would like to take a word.
Well, certainly, without question, the research process is clearly a strong collaboration between clinicians, clinician investigators, patients, and scientists such as Raj that are really looking at the mechanistic science, trying to understand the biology of the disease. I always view that it's kind of multiple streams of research in parallel, with many labs around the world looking at different aspects of this. But certainly, the work you presented is very intriguing, and certainly, very important to study. But, you could think of the scientific process are multiple things kind of being tested in parallel as opposed to sequentially. Not that one scientist exhausts their ideas and then another scientist, kind of picks up that baton. It's somewhat of a kind of a marketplace of ideas as things get tested. But you're right, the more basic the question in terms of earlier in the biology, sometimes the more challenging it is to obtain funding. But let me hand it off to my colleagues here who, again, we all both collaborate with similar individuals, but also with different individuals in parallel. So Marina, of course, we're part of the MPN Research Consortium, which is a collaboration itself between laboratories and our clinical research. Marina, why don't you tell me a little bit about the consortium?
Good morning. So actually, exactly that's what I was thinking about as well. So I'll start with the MPN consortium. So this is a collaboration between multiple basic science researchers. Right now, it's a group of institutions in the U.S. and Canada called MPNRC, MPN Research Consortium. And the idea is that, the only way we can find treatments and cures for these diseases is if we have many people working together on different aspects of the disease. And so what we have is a number of institutions, a number of individuals who are doing basic research in laboratories, and it's based on also mouse models, like here, as well as samples from patients with MPNs, looking at different targets in the laboratory, and which generates idea for clinical trials that are also run by the research consortium. And so the way that we work is that the ideas that are generated in laboratories then lead to clinical trials where we test the ideas, and then as we get samples from patients as they're treated with whatever the agent is, then the samples also go back to laboratories where we are able to further study what happens in the cells, and how does it relate to what happens in the patient, and then further ideas are generated. And so, I think a lot of the agents that are currently in clinical trials that are sponsored by industry initially were generated from laboratories of people working in this consortium. And this is really how we move this field forward. And I think looking at the work here, very, very interesting, intriguing ideas, again, where we're taking mouse models and patient samples, testing different hypotheses, and then potentially coming up with a target that could be used as a medication, as a drug. And potentially, this is just one of the pathways that's important in MPN pathogenesis, in how the disease how disease happens in stem cells and how it results in the disease phenotype that we see. And typically, what we are realizing now is that we need to be able to target the stem cells where this process happens. We also potentially need to target the environment around the stem cells, which is typically the bone marrow itself. And probably the combination of these targets is what we need to really get to the root of the disease. And I think here what we saw was really intriguing work looking at metabolic pathways, which are clearly important in many different cancers, including MPNs. And this is a very important question because as a clinician, a lot of patients will ask me, what happened? Like, why did this happen to me? Is it something that I did? Is it something that I'm doing? And my answer is always, it's usually multiple factors that's involved, and it's definitely not something that you did or something necessarily that you're doing. It's just a bunch of different factors that come together, and that's why this disease happens to you specifically. And when patients ask me about diet, my answer is always, we don't know what is specifically the best diet for each individual patient. But the usual answer that I think is the best answer that we know right now is you need to have a balanced diet, right? Mediterranean-style diet. I don't think that we necessarily recommend that patients restrict themselves to a very specific diet, but just a balanced, healthy Mediterranean-style diet is what we recommend for patients with MPNs, and I think in general. So I think what's important to remember here is that, I think like you said, mice are not little people. And certainly these mice that are genetically altered are also not exact models of the MPNs. So what we see in these mouse models, and specifically with very restricted diets, is it gives us a hint of what we should be looking for in patients, but it's not a direct pathway to the patient. I don't know if I'm making sense here.
Yeah, perfectly said. Morning, everybody. I'm really happy to be here today again. I'm from Houston, Texas. So I was looking at your fantastic presentation. I was commenting that everybody in Texas would have MPN with the amount of fat people take. So there must be something wrong with the idea completely. But that's not correct. MPNs is such a complex disease with the integrated inflammation that might start way before we do see the disease, where a lot of, as we were heard yesterday from Dr. Mesa and everybody, patients with very early disease suffer tremendously with a lot of symptoms. They are clearly tied to the underlying inflammation and the process that may have started way before that nobody was even aware of. And that gets integrated with the whole cardiovascular risk, the thrombotic risk, the serious consequences, and so on and so forth. So therefore, it's tremendously difficult to develop a model that would work generously for everybody and for each single situation, because once we start looking at one model, then the next from the different mechanism filled in, and that's how it is in cancer, but that it is in the whole medicine. It's just the human body is just more complex mouse, pretty much. So that speaks about although we from US and we trying to collaborate from the research, the scientists, to the bench, to the patients. I do believe that these collaboration need to be more global and the patient basis. I'm really super thankful to Peter, the global MPNs and all of these efforts, because we have to more include people from Europe, from Asia, from Africa, from everywhere, where they don't have the resources, nor they have the support from the companies or the governments to include, and they just suffer. And we could learn as a group and as a collaborator more, and there we really are very appreciative of our patients' advocacy and people like Peter and everybody. They do tremendous work to help us connect together and answer the questions that we see as a clinicians, but we see from the other side of the bench. So it is really important to us to raise the voice and tell us from your perspective that this is what is lacking, these are the answers that we don't have, and this is what we should be asking, and then we'll be, super happy to collaborate with like Ray and everybody else to really move on the needle in the research that goes more with the clinical needs. That's I think is very important for the whole community. I'm going to pass it along to Israel.
Thank you. I echo everything's been said, and just how important these meetings are. Just talking to Rao earlier this morning, and I'm going to bother him with a whole bunch of questions during the break. This is how the collaborations are actually formed, believe it or not, in sort of informal circumstances and events like this. I'd just like to add sort of an anecdote, which doesn't have anything specifically to do with MPN, but I just think it's interesting, sort of food for thought, excuse the pun, which you weren't able to do, Rao, because you fed your mice chow, and then you fed them high-fat diet, and the mice were sacrificed. I came across a very interesting study, which wasn't in the MPN field. I'm not sure if anyone came across it as well. I think it was published in Science or one of the science journals in the last couple of months, where researchers similar to Rao, they fed their mice chow, which is regular mouse food, and then they fed them like a McDonald's-type diet. They fed them a junk food diet for about 2 or 3 weeks. And of course, they put on weight and they became chubby and fat like you showed us. But then they didn't sacrifice the mice. They took away the McDonald's food and they gave them back the chow. And you know what happened? The mice wouldn't eat the chow. They literally starved themselves almost to death. So that's food for thought about what a bad diet can do for you.
Absolutely.
Okay, I don't know from the industry people, which is here. I don't know if anybody is from a medical department. Otherwise, Anna, would you like to?
Artur,
Yeah, Antonio, sorry.
Arturo Molina, Chief Medical Officer at Protagonist Therapeutics, and we know Dr. Rao pretty well. We're actually collaborating on a project that has not been disclosed, but at Protagonist, we're developing rusfertide for the treatment of polycythemia vera, and in the process of doing that, we've learned quite a bit about MPNs. I cannot talk about the project right now, but I can say is that we've had a very fruitful collaboration, and now recently we've engaged Dr. Ron Hoffman as well to work and brainstorm on a potential treatment that we are working on that needs further fine-tuning. Sometimes when you're discovering or developing new drugs, you start out with a tool compound where you just want to make sure you're getting the target, but it might require doses that are too high, and then there's the optimization phase where then you optimize the compound so that you preserve the targeting effect that you want, but at doses that are likely to translate into something you could actually give patients. So stay tuned, and I just wanted to recognize Dr. Rao as well, because he's been a wonderful collaborator, and we will continue to work on drug discovery efforts in this field.
So basically for the industry, the cooperation with persons with Rao is vital, because this is where you get from your new products. Is that right?
A lot of the new products do come from collaborations. Some of it is external innovation. Some of it is collaborations with academia. And that's one model that has worked very successfully.
Anna, as you have here, I must have you talking. How is that happening in large companies? Are you also, is your medical department, research department, do they have a lot of contacts with people like this, or is researchers' work done by ROA, is that also for a larger pharmaceutical company, a source of information to develop new products?
So I hope I can explain good in English. I think our medical department are moving forward, trying to make more collaboration with people as him and related with studies, take into account more things like nutrition and habits to increase the quality of life of patients, to make like a whole approach of treatment. But it's difficult for us to get or to give support, financial support to those investigators, but we are trying to do with a global medical team.
Okay, thank you very much. Just behind you.
No worries. Hello, everyone. I'm Thomas from Morphosis, as Peter said, and which is a biotech company, and we acquired, for example, Constellation, which was an even smaller biotech company who invented or researched the pelabresib, right, which is now, had amazing phase 3 data, and which we developed in the phase 3 trial. And now we got acquired from Novartis as they hope to bring this to market. And my point I wanted to make is that I think it's a whole ecosystem where everybody has a role to play, right? And it's academia who is very early on. Then you have like smaller biotech companies who partner or like who outsource. And fun story is, for example, with pelabresib, 12 years ago, when it was not even in a phase 1 trial, Constellation, got funding from LLS, which is the US patient organization that has this wonderful TAP program where they, also invest and co-invest with the biotech who are looking for funding. And I thought that was a wonderful, wonderful program to help also, make connections, as we're doing here, to co-fund innovation, and they have been a partner to us ever since. And so that's why I think, it's not right or wrong. It's like, as I think Ruben said, it's also agile, which I love. It doesn't have to be like this like very old school process. Yes, we need to follow a process, but I think it's very, very good and I think will advance science if we are doing this in parallel and be as closely connected as we can be and to make those connections.
Absolutely, Thomas. Mm-hmm.
Now, from the patient side, I just would like to know, you understand the importance, of the work which we're doing and how, if we as patients work closer together, how can we help that, Rao, he can continue his work, and as you see, he works with a lot of blood samples from different patients, which today makes life a bit easier. But I think, we as patients, we somehow we have to stick together and give him the real patient information. How do we feel? How are we reacting on it? Somebody wants to comment on that?
Yeah. So me as a patient and working with you in the association. First of all, thank you for the very educational presentation. I think it's also needed. For me, it's connecting the dot. I think was yesterday Ruben Mesa mentioned the misalignment between what patients are looking for as priority and he saw clearly that patients are saying, okay, symptoms treatment is okay, but we want more. No, we want to see that someone is looking for the basics, so for disease modification. So I think as a patient, and confirming what Ruben Mesa mentioned yesterday, we need to see that someone is working on this. I think it's needed. It's needed to see. It's very appreciated that we receive educational in this sense. So I think voice of patient for me is to confirm the misalignment and the needs of seeing that something is happening on modification disease, going to the fundamental, solving the basic questions, which are the difficult ones. Why this is happening? And so I think it's super important that this part of the community exists, no?
Well, it is. I mean, the problem, but how do we bring? How can we help? But all the work which is doing, but it's finally it ends up to our pharma industry, who then can work a new product out of it.
I think what we're doing here, so giving them voice, I think is for sure relevant. So I think patient association can play a role here, giving voice to this part of the community, and so the other part of the community listening and see the opportunity, and maybe we have to connect the dots. So that's one part, and of course the difficult one will be, helping to the financing, to the allocation of the finance thing, and making this flowing in this direction. But
John, do you think that our project MPN World could be a tool to bring that forward?
I think there are two things to say. The first is the role of patient and patient organizations is changing for the good. I think in the last 10 years, in the beginning, we were listening to what researchers were telling us. I think we're slowly growing to a situation that researchers are listening to what we are telling them. Yesterday, one of the slides of Ruben showed that many patients are more concerned about progression than the actual symptoms they have. And if you see the potential of GDF-15, for instance, this is typically something that patients are waiting for. Not just to don't have this painful biopsy anymore, but if this would be an indicator of progression, this is what the majority of patients are looking for. So it's our role to push the research community and the pharmaceutical companies to invest in this kind of solutions. That's point one. The second thing with regard to MPN World, I think by bringing all these patients together, we will have a stronger voice. But not just that. If we have enough detail of patients, then we also provide you a pool of patients in a far more easier way to make selections and make it easier for you to approach patients. I know all the limitations and the legal problems that can be in that way, but there are solutions for this. And once we have this pool of 10, 15, 20,000 patients on this platform, it makes it easier to do a proper inclusion filter and get the right patients for your research.
Absolutely.
So that is one of the roles we want to play as well.
Mm-hmm. Thanks for bringing the second point. Definitely looking more and more collaboration from my side, especially patient samples, as Marina mentioned. I'm very open to collaboration. I already talked this part to Ron Hoffman, very good discussion and in good terms, and also with Professor Ruben Mesa. I'm very close to what he's doing, and with this expertise, I would like to also team up with people like Ruben Mesa and Ron Hoffman, and my former mentor Rageswara, who is now in the US. So they are a bit far from me in Europe, but I think with this, I'm good in connecting in one way, but still at the finance side in Switzerland, I think it's very expensive. The same research you can do 3 times more in Germany, even the neighboring countries, and import the salaries and everything too expensive to do the same research. So if anyone is interested, I'm happy to discuss, collaborate companies and laboratories and foundations. And also, if you come across anything around here, something, I'll be happy to sneak in and you make use of it for a good reason. Mm-hmm. Thank you.
Please. One second. Put your headphones on, please. English speaking.
Sí.
Okay.
Okay. Bueno, mi nombre es Cristina, yo soy paciente de PV y en lo que yo demando, y creo que lo hablamos en la última reunión en Valencia, es que no vayan por separado la hematología de los problemas cardiovasculares o el cardiólogo, o como en este caso no vaya fuera de la enfermedad la nutrición. Creo que está poco estudiado y que es como que al final cada uno va por su lado, pero no nos ven. O sea, yo lo que le he dicho hoy de la alimentación, evidentemente sé que la alimentación es importante para todo, pero creo que en España por lo menos no se tiene en cuenta. Y además lo digo de primera mano porque mi hija ha estudiado la carrera de nutricionista. Las prácticas las hizo con un pediatra de tutor, porque no hay nutricionistas. Y cuando uno va al hematólogo no le derivan al nutricionista o al cardiólogo para que te haga ser consciente de que no solamente es el medicamento que te baja los niveles, sino que tienes que aportar mucho más a todo eso, y eso está en la parte del paciente, no está en otro sitio. Entonces, bueno, eso era mi demanda, ¿no? Que creo que se nos debería de ver como un conjunto de situaciones, no solo lo que es cada especialista. Uno mira el hemograma y de ahí no sale. El cardiólogo te hace las pruebas de cardiología, pero tampoco te indica, mira, esto está mal en la analítica, te voy a derivar al nutricionista para que te ayude a controlar todos estos niveles, y creo que sería una parte muy importante. No sé cómo lo veis.
Estamos viviendo en un mundo de especialistas, ¿no? Cada vez los médicos son más especialistas. Cada vez hay menos médicos que quieren ser médicos generales, porque las condiciones no son muy buenas, especialmente aquí en España. Y también luego depende de la enfermedad. Si tú tienes una enfermedad de corazón, te cuidan mucho más en todos esos sentidos, están mucho más organizados. Yo lo he visto porque en mi edad, cuando vas al médico te encuentran algo. Yo no puedo ir al médico porque es algo pagado. Han encontrado algo de corazón y ellos tienen en el hospital del día, pues tienen un grupo que es mayormente un médico mayor, que ya no está en las consultas, y ellos te cuidan. Ellos te hablan de todo esto, de no fumes, no bebes, en fin, todo lo que no puedes hacer. Al final no preguntas por qué vivo si no puedo hacer todas esas cosas, ¿no? Pero ellos realmente ahí tienen un seguimiento. Esto realmente en las enfermedades raras existe muy poco. Lo que, por ejemplo, nosotros en la fundación lo que hacemos, tenemos un programa con algunos hospitales en España para crear una enfermera de enlace para los pacientes de MPN. Vamos a tener dentro de poco un programa de piloto en el Hospital del Mar de Barcelona, y que vamos a poner una persona que estará financiado por la fundación. ¿Por qué? Porque, bueno, todos sabemos, si queremos llamar al médico, es que no podemos llamar al médico. Y esto es un problema no solamente de España, sino fuera en Europa, en todos los sitios es igual, ¿no? Entonces, ahí pues esperamos que podemos dar esta información, que esta enfermera luego ya hable más de la nutrición. Ahora bien, con mis charlas con los médicos y cuando estamos con Genfin y con esta gente, la mayor parte de los médicos están ya pensando en esto del comer, ¿no? de la dieta mediterránea y esto, ¿no? Poco a poco está viniendo, pero es que es un problema de las enfermedades raras, es que no falta la infraestructura realmente para ayudar a tan poca gente, ¿no? Y si vamos a niños es peor todavía, ¿no? Porque todavía somos menos. Y ahí somos nosotros como organización que tenemos que ayudar y tenemos que dar el apoyo y la información a los pacientes. Luego el problema es que muchos pacientes no quieren seguir lo que estamos diciendo. Así que es al final siempre un poco. Pero lo que has dicho tienes toda la razón. Estamos totalmente de acuerdo y yo sé que en Holanda hay problemas muy similares, pero ahí ya, digamos, todo el movimiento de pacientes está mucho más avanzados que nosotros y ellos, tú ves en sus páginas, en sus sitios, tienen mucho más información sobre nutrición. Nosotros mañana, por ejemplo, hablamos de nutrición y tendremos un especialista aquí a ver que nos cuente, ¿no? Aunque realmente la tenemos delante de la puerta, que es la Mediterránea, ¿no? Y yo he sido el primero de aprender que es una dieta mediterránea, ¿no? Pero es verdad, al fin esta coordinación estaría bueno, pero vendrá, lucharemos para esto, ¿no? Bien, había alguien más que quiere hablar, ¿no? English. Oh yeah, I thought of somebody else now who wanted to talk. Yeah, no. What about how do you do that in France? I know you have your group is very young. Vivre avec une MPN, Karine, qui est which is very active. So I'm switching to French. And you have a very big support from Jean-Jacques Kilijan, which is a big advantage. How do you see this? You know, what we're just talking now, about information from young researchers. How is it working in France?
Well, it depends. Haruko could speak about Dr. Kalijan in Hôpital Saint-Louis because he is a world-leading expert in that domain. So we, as MPN patients, we also have the opportunity to meet with high-level scientific experts in our field, because they are clinicians, they are doing clinical research, but they are also doing basic research. There is one point that we need to further discuss, I think, is yesterday it was mentioned that we could create a registry of patients to follow up the outcomes and so on and so forth. But what is important for basic research is to create a biobank. So we are used to give our blood to check the counts. But if we give your blood to a biobank, then it will serve to basic research whenever they need to check with some hypotheses. So this is something that we should keep in mind. And that I will discuss with my physician next time I will see him.
Yeah, it's interesting what you say about the biobank. Some of you know I have another foundation in Switzerland, which is composed by the most important hematologists, MPN specialists in Switzerland, which is easy because Switzerland is so small. It's we have all the different cantons where you have your specialists. So we formed the foundation 3-4 years ago, and the idea is, obviously also a register, which we have in the meantime. We have a registry with the analytical data of the Swiss MPN patients. And every hospital is using the same data sheet, which I think is very important. In most of the countries, every center, every hospital still has its own data sheet and, looks a little bit different analytical data. We managed to do that in Switzerland also because it's small and this is how we did it. And some of you may know the name of Rageswara. He worked many years with Professor Rageswara in his biological department. He was part of the group of physicians who discovered the YAC2. And he is now, he in Basel and in Bern, and in Zurich, we do have biobanks. And as Radek is leaving, all his department will be closed. So, we have a biobank which is existing since 15 years approximately. It has a big value because, all these samples are there. So we are, at this moment, we are negotiating, but this biobank could come in the hands of our foundation. I tell you, the figures we are talking about, it's expensive. And Switzerland is the double expensive. So it's a question of money, but it's absolutely right. I mean, this is one of the important points, that we have biobanks where we can go back, I said, he can get blood from hundreds of patients and the patients don't have to go to him. So, but he gets these samples. These are all coming basically from biobanks. So it's a biobank is something extremely important and simply increases the work which we have to do in our international organizations. We have to provide this. I fully agree there. But it's a very expensive item, and money is not so freely available. It's very good. Thank you. You fight. Who wants to first?
Okay. I have a question to Rao. You said platelets feed on fat, and I have an ET, so would it be rather wisely to cut on fat than sugar? And otherwise, fat and sugar, they can be transformed into each other. So second question, how does this Mediterranean diet work at all? Because it's quite fatty.
So it's a very, very good question, and actually both questions are very good. I didn't get the time to go into detail. How metabolism, like cancer cells, metabolism also so flexible in general, even when you're healthy. Okay, if you provide more fat at the end, also it can convert in the liver to carbohydrate again. Okay, so but that is depend how long you are starved from the glucose or the fat. Okay, but it is entirely even different, even more exaggerated, this reprogramming of metabolism when you have a MPN, like MPN or cancer. So now you're asking question, Rao, if I take fat, if it convert to glucose or sugar, so it doesn't matter if I eat directly the sugar, right? I'm not saying here you should not cut off all. You really reduce, if you have a specific preference, I love Swiss cheese and I like to eat only Swiss cheese, yes, it has consequence on over time on fat, on platelets, for example. No, I don't like fat, but I like Swiss chocolates more, and I only eat Swiss chocolates, then probably it will, long term, it will impact. So what I'm trying to say here is be mindful what you are eating, how much you are eating, and that also, a lot of research needed. These are the kind of preliminary primary insights. Again, as Marina said, it's from mouse models. The people started looking in patients, okay? But you need to have that idea definitely should come from the lab, okay? Clinicians, what they do, all the findings, what we do in the lab, they transform, they use that provided by a pharma company. Yes, they test, okay, how much to test, how long to test, and then take the blood counts. But the primary idea often in our community is not recognized. In other words, basic scientists are kind of second or third class citizens, I can tell you. It means my little experience, you call it frustration or problem, but it is not often recognized as it should be. Right? Because if you don't have idea how pharma can develop a drug, how a clinician can test it, as Professor Ruben Messer said, we need to understand more biology first. Okay, 20 years now, we know the mutation, and clinicians say, yes, your blood counts are higher. I switched from Ruxo to interferon because clinical trial said, yes, we are using mouse models. I don't have degree to work on patients. Okay. But if I have the same degree, I can also test the same in the mice, what I test in patients. But clinicians have that opportunity, for example. But clinicians should know the biology, right? You need to know both, right? I cannot do everything, and they have a right to do something directly with the patient. So in other words, you need to consult your cancer nutritionist who is well educated, who is teaming up with your hematologist, like colleague said. You guys have to work together in the clinic, right? And they are advocate enough to guide you how much to eat, what to eat if you have PV or ET. Again, what I told you, just take as a basic information. So we are getting there. So these kind of trigger people to think, okay, that study showed, maybe somebody else shows something else. Okay, combine those two and then bring to the clinic and then eventually go to. So answering your question, how much to take, what to take, it's very premature to say. But be mindful not to take too much the same thing. Okay, so that's the message I can give in a fair way. But it's a very important question you raised, yes.
Let's go to Thomas.
I just wanted to answer a previous question, what we as patients can do and just recall that in Germany we have a bioregister for the MPN patients where all the samples of, I think it's 2,500 patients or 3,000 in the meantime, is located in one city, and they can use it then for that, for the studies and the science.
Mm.
So thank you. I just wanted to add collaborations, in terms of collaboration. I have very good experience working with the states and Canada, and it's very difficult to get samples from Germany and France. And the most hardest I found is from France. The most easiest and flexible is from United States. I had a good experience also from Toronto. I had very good access to the sample. The very hardest I found, I tried many times from France. In part, probably cultural things, but also the administrative stuff. So they need to have something needs to be approved, whether it can be shared or not. So that is one of the primary hurdle, and also academic competition. People like me, no one. If I come and present, no one cares. The same thing presented by Professor Mesa, for example, and it's like a Nature paper, right? So it's more people read it. Right, so those differences I think we should ignore. We should ignore in terms of we should focus on the true science, and I think that is very, very important. As a biologist, I'm not self-centric. Both should be important. Fundamental discovery and the clinical investigation, both are very important.
But you see what Thomas says. It's one of the problems which we have in Europe, not only in the bio. You have a little bit here, you have a little bit there. You have it in France, you have it in Germany, I'm sure we have it in Italy, we have it even in this small Switzerland, we have one. But they just keep it for themselves. And he's right. I mean, if I'm going now to ask, it's probably under GSG, who has the biobank, it's very difficult to get a cooperation. And this is our problem. We would like to have more information. And this is why I'm trying to create here a group of patients that you can help us to have influence in the local organizations. If you biobank in France, I don't want to have a biobank, but at least get information from there. Get details, get backup, which we then can put then on disposal of these people. So we would like to be like a catalyst, where we really can bring the things together. And why are we so strong in the States? Most of the money is in the States, and they are much more cooperating to each other. They also have different centers, but you can see, I mean, here in the backside we have 3 persons from the industry, American industry. And, we're friends, we know each other, we work together, we exchange information, we exchange results. And this is what we're not doing here in Europe. Where there is, Chemfin is not working with GSG, and, and it's Europe. Everybody has his own history and wants to keep things for himself. This is, I mean, I remember when we were talking about to getting the data from Chemfin into a big organization of Harmony, and they said, no, we are afraid, because if we give it to Harmony, Professor Barboui, he can look at it and he can see our results. We don't want to have that. It's, we have a philosophy still in Europe, based on the history which we have, unfortunately, which makes this life a bit more difficult. But anyway, many hands up now.
But maybe there's a point. It's where we, as patient organizations, can help, because it's nice that we have that 3,000 patients in that register study in Ulm, but compared to the number of patients we have with MPN in Germany, it's already a small number. So maybe things would just get better if we encourage the patients to really donor their samples for that register, so they don't have to behold it for themselves, because they have so much that they can give it. So maybe there's possibility if we encourage the patients to work with that registers.
Okay. let's keep up these ideas, and then we will try to work on it.
I just wanted to mention really quickly again, and I think Raul already mentioned it. So we, in the US, through the MPN Research Consortium, which is National Cancer Institute sponsored consortium of institutions that's run by Dr. Ron Hoffman, we have a large tissue bank of samples from patients, including sequential samples, and they are available for different researchers throughout the world. So this is, I think, a tremendous resource that is available for basic science researchers with patient samples. And so I think it's just a great resource. I just wanted to make sure that people are aware of it.
A researcher working in Switzerland, can he get
Yes.
cooperate?
Yes.
Yeah.
Thank you, Barry. So I had a very good opportunity 2 weeks ago to discuss with Ron Hoffman at an ESH MPN meeting in France. So that meeting went very fruitful, and Ron, I know him for the last decade, 10 years. But this last meeting was really changed to the collaboration side. I'm also hoping that I get some of those samples. Thank you, Barry.
You want to have?
About the question regarding diet specifically for ET, it was about 5 points ago. You know, it's as a practicing clinician, it's a very difficult question to answer. So I think we were discussing yesterday evening. A lot of our patients ask us, you know, what can I do? What should I be eating? How should I be exercising? What kind of lifestyle should I have? People want to be empowered, and they want to move their own care for their own disease forward. And I think that's laudable, and that should be encouraged, and I think we try and do that as physicians. But we're not quite sure exactly what to answer and how to answer the question. And one of the things that we have to remember is that the vast majority of patients with MPNs will have coexisting diseases as well. And in the Western world, a lot of these will be metabolic diseases. So there'll be high blood pressure, high blood lipids, diabetes, obesity, which is classified as a disease, and not to mention smoking. So I think, you know, we're looking at the basic biology of metabolism in MPNs, which is fascinating beyond. But in clinical practice, this has to be taken in a context of other health issues, which in and of themselves require treatment. So what I usually tell patients who are perfectly healthy, I say, you know, we don't really know exactly what kind of diet you should be on, just like Raul said, but probably a good rule of thumb would be to maintain as healthy a lifestyle as you can, because if you have a disease in a diseased body or a disease in a healthy body, it's probably easier to deal with it if the rest of your body is healthy, so to speak, and then you can focus on watching your MPN. But most patients don't have a healthy body. They have one of these other underlying diseases, most of which are metabolic, and so that's a big driver, at least for me, as to how to answer your question, which is a specific ET question. But it has, I think, in almost each patient, it has much more global health ramification. Unfortunately, most people are not healthy, especially older people in whom the MPNs are more common overall.
Thank you, Martin, for sharing this. I think it's very important. You also added the comorbidities. MPN patients often have atherosclerosis or cardiovascular symptoms. That's where actually also people now little bit moving direction. If people come to clinic with cardiovascular complications, nowadays they're also taking blood and doing the same NGS sequencing to see if they also have an MPN or JAK2 mutation, for example. That's how people actually finding a connection between comorbidities like cardiovascular and MPN. So in that situation, still, I think it's good to have a nice diet so you can reduce both cardiovascular problem and MPN. I think at the end you have to take a good diet. Mm-hmm.
Our Swiss friends are very quiet. Good.
I think it's very important that the patient organizations in all Europe or all over the world work together. They connect them, you make them with global, but if we in Switzerland to try to make connections with other countries, it's really difficult. It's hard because all the legals, all the rules, and I think every country should start at home to collect the patient, to know who is the patient, where are the patients, and then if we know that, we can bring them to the research, and it would make a lot easier. But we restarted now in Switzerland after COVID, and we tried to get to work with other countries, and I see there are some borders, and we have to move over these borders. And I think the first step is that we bring all the countries together. What you are doing, thank you very much for that. But if I'm here, I don't know if somebody from Italy, from Austria is here. I would really appreciate if it would be possible with the countries around your country work together.
Marcus, can I add one quick thing to that? I think maybe Professor Ruben Mesa and Peter, people like them, should initiate that these things should be done by WHO, World Health Organization, right? Because they are there, I don't know what they're doing. Whenever COVID comes, they pop up. When there's no COVID, you never hear WHO. I think they should invest. WHO, World Health Organization, for example, they should put these, and they should sponsor. I think people like Ruben and Peter should go to them and then influence such things. I think that's more realistic. Otherwise, if we fight and we talk about problems, we never get solution for that. I think if you have such big global organization, they should sponsor, then the foundation can run, and we will have a nice data set. Right.
Question, but if you don't have to go so far, the World Health Organization.
Or like, I'm just giving maybe any other equivalent one.
Well, we have seen in the recent examples how strong they are, in the war areas, how much they can do. But it is something, that money is available. I think, if we really manage to get the group together as John and me, we want to have it, we can present projects in Europe. The European community has billions available, and it normally goes to the wrong hands. It normally goes to the people who has enough already. But I got some contacts now, we're trying to work on it, and if we will be able to have a real project together, there is money available, and this is not 10,000 euros. It's really it's big amount of money, and but they want, obviously it's Europe, so they want to have information. If we bring a project, we need it all supported by all the different countries. I cannot bring it only, well, from Switzerland, be too small. They wouldn't look at it, and we're not in a common market. But even bring from Holland, from Spain, it's not enough. You really have to bring it together. This is why, for us, these meetings are so important, that we can work together. So, but we will definitely be done. I won't go so up like the World Health Organization, but on the idea, you're absolutely right. Now, I still would like to have one opinion of one person which is here, which is for me very valuable, which is Laura. You are one of our main specialists of MPN in Spain. You have a very wide view of what is happening. I don't know if you know Raul, but if not, you will have.
Now I do.
Now you can talk to him. You are in the middle. You are a physician who has every week, I don't know, 50, 60 MPN patients, which you're having. How do you see all that change? All what we are talking now this morning. What is your opinion about it?
Well, it's a big complex picture when we are talking, because there are many kind of different aspects. I think that it's true that all these kind of investigation, the biological, the clinical investigation, the clinical trials, they are different things, but they go together. And sometimes it's difficult to going together to this point, because what we want is to resolve this problem, but we need different people working in different areas. For example, talking from my side, during the week, I have no time to go to the lab. I just see patients. And all the ideas, because you need questions, that is what he said. We need questions to investigate, to know where start from or what to investigate. It's not, I'm going to investigate NPMs. It's that is, you need questions and you need to answer questions. And sometimes these questions don't, they can be born in the lab, they can be born in the physician office, and we need to work together. And I can do Ro's work, and he cannot do my work, and we cannot do the pharmaceutical work, because the day has only 24 hours. And this is quite important, because we are working on that, because we, for example, what we do in HEMFIM, trying to get this registry that is clinical information that biological investigator needs, that we need to find answers, different kind of answers. For example, maybe from these clinical registries, we find answers on maybe, I don't know, the dose of aspirin, or how well we do some kind of treatment. And these answers are needed, but we need also to collaborate. And sometimes it's not easy to work with the biological, the primary investigation. So we need to do the biological, the translational investigation, the clinical investigation. We need to do that. And for that, we need time and people. and I think probably that's where we have main problems, at least in Spain. But we all keep doing. And I think that the fact that we are all here on Saturday and trying to find out, this means that we really want to do it. We really want to work on that. And people like Roy, I'm for sure that he's always looking for the money for the investigation. That is really hard. And that also takes time, because I think that the best Roy can do is be in the lab thinking and working, not searching for money. So we need to find people that can do that work, because we have to do a lot of paperwork to get how do you say, Becca?
Ethics approval? Ethics approval, for example.
Sponsorship, that one. We need to do, for example, in Spain, there is a big kind of project, that is national project, that you can get money to do an investigation. But you have to work almost a year to fill up all the paperwork to try to get approved. And it's really difficult, because maybe there are 10 projects for whole Spain, for whole pathologies, for everybody. So all that paperwork, all this problem with financiation, I think this is something that there should be maybe a commission that you take your idea and they see how they can get the money to do that investigation, for example. I think the way we have to do these things to do investigation, clinical, biology, all these parts that are all important, is really difficult because of this, because of the bureaucratic problems, the paperwork, and kind of things. And that will help a lot to make things easier. And I think that people really want to collaborate, and we need to cross those boundaries that we are talking, that they are real. Europe is not one country, as US, it's many countries, and that makes things quite difficult. And for example, this that you were talking about, the harmony, this project, I have my doubts if that is the answer. I think that is one tool, one other tool, but it's not the answer to keep all this machine working. And I think that the role of the patients and associations that are starting to push to make themselves here, that is quite important because, for example, something that we have talked many times is that, for example, clinical trials, that you have many questionnaires, many work, but I think that patients should be involved in this kind of investigation more properly, not only as the people that is being studied. They have to be involved in the design of these clinical trials, for example. And a good thing is that to know that things are moving and where the problems are is going to help us to keep on researching and keep on resolving problems.
Sure. There are many movements going into this direction. You know, for example, I am a member of the one of the boards of Hospital del Mar. You know, and it's an expert group. They are every 2 weeks they are deciding which clinical trial will be accepted or not, and under which condition. And now, since about 2 months, I am assisting to these meetings as a patient representative. So, you know, they are trying to get my opinion as a patient, which is not always easy if we talk about a myeloma or we talk about the breast cancer, whatever it is. But still, you know, you can give a general information because many symptoms which we are having as patients, they are identical in many other illnesses too. So I think this is very valuable. I know also Vall d'Hebron is having an initiative to include patients into certain decisions where we are participating. So it's coming slowly but surely. And you know, also the things which we're discussing here today, we're still talking basics. We are basically below his level, you know, before he even starts with mouses. We haven't reached the mouses yet. So we're trying. Actually, what we're trying to do, or what I'm trying to do, and John is trying to do, is we're trying to find a common basis. You know, you're absolutely right. You know, we have a lot of obstacles to go through, a lot of them, not only internationally but also locally inside of Spain, for example. You know, you have different problems, but you know, let's take every problem as a challenge, and you know, we will be able to make it. Thank you anyway.
Okay. I just wanted to reply to Laura. From the association point of view, we are always trying to look for ways of connecting and doing things. And it came to my mind, I work in the industry, I'm an engineer, and we have kind of the same problems. Every day we have to do our turnkey projects and sell the products and keep the company going. But we also need to dedicate some of our resources to research and innovation, because that's what keeps us in the top of the technology and allow us to be on the forefront of the business. And we also apply for help to the administration for research, and we have the same kind of problem. You have to take people out of the computer to fill up tons of documents to apply for one of these helps in research. And we use the help of external companies that dedicate themselves to that they're very good at it. So maybe from the association, we could dedicate some of our funds and the money that we gather for these kind of events and for other activities, maybe to give you that support. If you are keen and developing something, you have your mind in something that you believe is going to be interesting, and you don't have the time to go to the lab, you don't have the time to go to the computer to type all those forms, maybe there we can help. We are not medical physicians or stuff, but maybe we can look for the resources if we don't even have that. So just keep that in mind.
So I was just going to build on a bit of what Peter had said, really regarding kind of your potential roles as patient advocates, really both with the healthcare system and centers that are dealing with both patients with blood diseases as well as with cancers. And I think this is more and more the standard and is incredibly helpful, from a range of perspectives. And sometimes perhaps, how we're using it in the States can be more proactive. So one, we have what are called patient and family advisory committees, and there are multiple of them that really help to provide guidance on a range of things. So for example, again, regarding actually care that is received. And again, you guys have a unique perspective. What is it like to go through a phlebotomy? How does one expedite the process of people that need repeated phlebotomies, or for receiving blood tests, or communicating around the results of blood tests? Two, clearly in the clinical trial process. Clinical trials have become exponentially more complicated. And I think something that has been helpful in helping to curtail some of that has really been patient advocate involvement, really providing a lens of really what is reasonable to ask people in terms of participating in a study. If the study says, oh, I think it'd be very nice if we had a bone marrow done weekly so that we could track the change in the bone marrow, wouldn't that be scientifically interesting? But ridiculous to ask an actual person to have to undergo those number of bone marrows. So these sorts of things. Oh, well, we'd like you to get the blood test done at the study center, on a daily basis. I think it'd be very nice to track the hemoglobin every single day for 2 years so that we could track that very nicely. So there's a lot of these things that come up that are absolutely ridiculous, but you really need the lens of, actual people that would need to go through it to really push back and kind of give that sort of perspective. And that's why, actually, Peter, your perspective, it could be disease independent. You may know less about breast cancer, but again, as an astute person who interacts with patients, again, you have a sense of, what is reasonable and fair and what is not. What I ask of people at our center is to use what we call the loved one standard. Meaning, if we would not want our own loved ones, my mother, my wife, my child, to go through whatever we're asking someone to do, then we shouldn't do it. And likewise, as we provide care, we'd want it to be at the level that someone that we love would have to go through. What compassion is there? Is the signage clear? Are people confused as to where to go? There are many things that add stress in the medical process. So all of you are clearly super engaged. Otherwise, you wouldn't be at a meeting like this anyway. So in many ways, you have the opportunity to really kind of drive that change, because many of these things in the States really arose from people saying, we want a seat at the table to be able to provide input in a range of ways. And having an MPN, you fall a little bit in between different groups. There's kind of one group that are patients with active solid tumors. And these are very different diseases, sometimes cared for in the same centers, but dramatically different in terms of the medications and things that are involved. Likewise, even in heme malignancies, the arc of an MPN is very different than acute leukemia or lymphoma, or multiple myeloma. So its own unique thing. But your guys' voice in many of these things is incredibly helpful, And if in your country this really hasn't arisen, that's where, again, I think kind of your national groups can really help to drive this. And sometimes just doing it well at one center, as an example, helps it spread to the other centers. Because sometimes once people have seen what is that impact at one particular center, then it's like, oh, well, boy, that was really helpful, and then help to extend it. So Peter, thank you for doing that, because I think that can help to impact a range of things. But those in which you find that there's less of that, certainly, be those advocates for change.
Well, I can't do it alone, so I need the help of all of you. It's a big burden. By the way, if it's short, we will have a coffee break. Professor Mesa, he will leave us at 12:30. So, if any questions you still have to him, grab him before he leaves, yeah. So it's, we're having, we're having a coffee break at 11:45. Please.
I just want to say that for all that money, Peter, you said that has the European Union that sometimes going to the wrong hands. Sometimes, as not always, for the high-level politicals, we are numbers. So we need to be the largest numbers as we can, as association, because that means votes. So I think that it's really important to grow every association, because those numbers can decide. They agree or not agree with our positions more than if are right or wrong. So I think that that's important, because it can make them decide to support or not, more than the rights, just the numbers sometimes. It's sadly, but I think it's true.
There's no question about the number is important, yeah. And we definitely need the cooperation. But I want to give you an example. We are now in Facebook in Spain. We are 1,000 members, yeah. We have about 350 PV patients. You know that we are in a battle with the government to get one of the pharma products released, yeah. And I have been many things to try to get it released. We even got an Italian professor flying into Madrid explaining everything. I have asked in Facebook, everybody, to every PV patient, please send me a mail and complain about that you don't have that product. I will collect it, and with all your letters, I'm going to the ministry. 3 replies. 3 out of 300. And that hits you, I am going to every autonomy which we have in Spain. I talk to the top sanitary people, making trips. I make 1,000 kilometers a day just to see people. We make it virtual. I went to Madrid to the Congress to talk to people, which was excellent, So we're doing a lot of efforts, but when you see, well, it looks like the patients don't worry about it. It's, you can do whatever you want, I mean, here you can see that we have many people who is ready to spend the weekend and to talk about these things. But very often the reply which you get, from the community is very limited. Holland, for example, is different. Holland has a different mentality. The patient has a different mentality. And also we have a problem at PV. Many patients, they don't have symptoms. They have, quiet life. So they're not going into the page of MPN. So, there are a lot of reasons for it, but sometimes it's very difficult. So we start already in our own organization with a problem of motivation. And I'm pretty convinced that exists in our countries, probably the same thing. So, but I think also there in between of all of us, we have to stimulate people to be more cooperative. So, anybody else?
So, a few things. I have worked in pharma for many years, more than 20 years.
You have to eat it.
Yeah. Well, a few things. We discussed about collaborations with scientists and so on. Well, I have to say that pharma is huge, it's big. So for you, things like that, I can see that maybe in preclinical, clinical, they will do a collaboration with you. But it have to be a reason as well behind why do we want to collaborate with you. Sure. It's not only because, well, we see that it's good for the patients, but also what is in there for us. And it's awful to say, but it is what it is. Then I have seen as well a shift from what they believe the patient wanted to, okay, we cannot do anything without the patients. We have really to hear them. What do they want? So the quality of life is really important, is crucial. So you mentioned about nutrition, and I think that more and more they're looking at not only we provide a treatment, but what is more for the patients? What is the holistic treatment? It's not just because I give you a treatment, but if you are having a diet that is poor, the treatment is not going to work the way they want the treatment to work. So I think in there is a lot of opportunities for collaborations with pharma industry. Then we discussed about the data. I mean, biobanks, there are fantastic biobanks in the Nordics, but then you have to go there with a protocol and a study. What do you want to study? How do you want to do it? But you don't have the money. So you know the protocol and everything. Again, you need collaborations. You need collaboration with a sponsor as well. How do we do that? So my point is, I think we really need everybody involved. It's just the pharma industry, the physicians, of course, the patients as well, to say, okay, this is really what is important for us. And we are going from, what was important was the survival. Okay, we give you that and you will survive 5 years. Not anymore. We want more. We want quality of life. It's not about how many years do you want to live. It's how do you want to live these years as well? And how do you have this voice to say, okay, that is enough. It's not only this. I want to really live well. Association with Europe, great. It's true, numbers. But we're so different, and we have the DPR as well, and that is a huge thing when we're trying to get data. And today, data is gold. So you see all these breaks that they put because they don't want to provide data. They say, what do you want that? Because it's money at the end of the day. So we really need to think how we can maybe learn for sharing experience from other countries like the Nordics. They have fantastic biobanks in there, and they are happy to share the data. Like how we collaborate together with more associations in Europe. And pharma, of course, we need them to sponsor the studies, but we have to go with a narrative of what is in there as well for them to work with us, because obviously it's a benefit for all of us.
Exactly.
Okay. It was a good, coffee is coming. I think it was a very good discussion. I'm very happy about it. So, anybody else has a question now? So let's go to a coffee break for half an hour, and we come back with new forces, yeah? Thank you very much for participating.